Over an aerial view of the Life Sciences building, text reads, University of Dundee, School of Life Sciences. Disease insights from stem cells. In an office, a small group of scientists converse. One of the scientists points to writing on a whiteboard.
Stem cells are an important tool for scientists to study how humans develop and how this goes wrong in disease. Here in the Medical Research Council Protein Phosphorylation and Ubiquitination Unit, or MRC-PPU for short, we use these cells to try and find the causes of human developmental disorders, such as intellectual disability.
A labeled microscopic image of a mouse embryonic stem cell. Scientists work in a lab, one of whom is injecting liquid into tubes.
Intellectual disability is characterized by impaired learning and social functioning, which affects an estimated 1 to 3% of the world's population. However, the causes of intellectual disability remain mostly unknown.
A scientist works in the tissue culture suite. She retrieves a dish from an incubator and places it under a microscope. She studies the dish.
Let's visit our tissue culture suite and see how we use stem cells in our experiments. First, we must ensure that stem cells are in the right environment to grow healthily. Like ourselves, they need warmth, air, and nutrients to survive. Warmth and air are provided by cell incubators and nutrients by a liquid soup. We regularly check that the stem cells are healthy by using a microscope.
A grey, labeled, microscopic image of mouse embryonic stem cells. A scientist works with the dish within a microbiological safety cabinet. She pipettes a clear liquid from the dish and replaces it with a pink-tinged liquid.
Then, before starting an experiment, we first count the cells to make sure we have enough. We do this by washing off the nutrient soup they grow in and add an enzyme to lift the cells off the dish.
The scientist pipettes the cells into an automatic counting machine. She then pipettes the cells into a 6-well plate and places it in the incubator.
The cells are counted using an automatic counter, and we take the number of cells we need to start our experiments, which is normally at least 1 million. Cells are then returned to the incubator to grow.
Grey, labeled, microscopic footage of mouse embryonic stem cells growing over time. The scientist removes the pink-tinged liquid from the 6-well plate. She pipettes the fresh pink-tinged liquid into the 6-well plate and returns it to the incubator.
The stem cells we study are from the embryo, which means that they have the ability to become any cell type in the body. We can treat stem cells with various chemicals and factors to mimic the environment within the body. This allows them to start to develop into specialized cell types that make up our tissues.
A colored, labeled, microscopic image of neuron cells from the brain. Labeled microscopic footage of cardiac muscle cells from the heart beating.
For example, neurons from the brain, or cardiac muscle cells from the heart.
A scientist extracts pink liquid from a 6-well plate, and then adds a clear liquid into a tube. She scrapes one of the wells, then pipettes liquid into a small tube and then places small tubes into a centrifuge. She pipettes liquid from a 96-well plate. She holds a container, which contains red liquid and a white membrane sheet. She shakes the container slightly, so that the red liquid moves over the membrane sheet.
We can then use these cells to study how tissue development is controlled, and how this might go wrong in people with developmental disorders. To unravel the communication systems that control tissue development, we must break open the cells to find out what is happening inside. We can then separate and collect the cellular instruction manual, DNA and RNA, and molecular machinery, proteins, from inside the cells.
The scientist pours the red liquid from the container into a tube, then seals the tube. She pours clear liquid over the membrane sheet. She removes the membrane sheet, covers it in a plastic film, and then places it into a machine. The machine screen shows a gray image of the membrane sheet showing lined columns.
These parts can then be analysed using different experimental techniques. This reveals how tissues develop, how this goes wrong in people with developmental disorders, and how we might better treat these diseases.
A black letter W appears over text to form the Wellcome logo. Text below reads, supported by the Wellcome Institutional Strategic Support Fund awarded to University of Dundee. A black and blue symbol, with the letters UKRI, appears alongside text reading, MRC protein phosphorylation and ubiquitination unit. A blue and white crest appears beside blue text to form the University of Dundee logo.